Hello there, and welcome to Sleep Science, where the mysteries of existence become gentle pathways to rest. Tonight, we're embarking on perhaps the most extraordinary detective story ever told: how life itself first stirred on our planet. Before we begin, feel free to share what time it is and where you're listening from. It's always wonderful to know where these quiet explorations are drifting off to. We'll be wandering back nearly 4 billion years to a time when Earth was a very different place. No trees, no birds, no gentle waves lapping at familiar shores. Just a young, restless planet where chemistry was about to perform its greatest magic trick, transforming lifeless molecules into something that could grow, change, and eventually dream. We hope this journey isn't too fascinating, because while we'd love to spark your curiosity, our real goal is to help you drift peacefully towards sleep. But if you do happen to stay awake a little longer, at least you'll come away knowing how a handful of ancient molecules learned to dance together, creating the first whisper of life in an ocean of possibilities. So settle in, close your eyes if you like, and let's take a gentle wander back to the very beginning of our story. Because every heartbeat, every breath, every thought you'll have as you fall asleep tonight traces back to those first tentative moments when chemistry became biology. Picture a world that would terrify and astonish you in equal measure. Earth, 4 billion years ago, was nothing like the gentle blue marble we call home today. It was a planet of fire and violence, wrapped in poisonous clouds and pelted by cosmic debris. The sky glowed orange-red, choked with methane and ammonia. No protective ozone layer shielded the surface from the sun's harsh radiation. Lightning crackled through the thick atmosphere almost constantly, as if the planet itself was having an endless, spectacular tantrum. The ground beneath was equally unwelcoming. Volcanoes erupted with casual frequency, spewing molten rock and toxic gases into the already hostile air. The surface temperature could melt lead in some places, while other regions were flash frozen by the planet's erratic weather patterns. Massive asteroids and comets still bombarded the young world, each impact releasing the energy of millions of nuclear bombs. It was the tail end of what scientists poetically call the late heavy bombardment, though cosmic shooting gallery might be more accurate. Yet, amidst all this chaos, something quietly extraordinary was happening. Water was beginning to settle. Not the clear, refreshing water you might sip before bed, but a strange, mineral-rich brew that tasted of metals and sulfur. These early oceans were hot, acidic, and absolutely nothing you'd want to swim in. They were more like a planetary chemistry set, bubbling with dissolved iron, hydrogen sulfide, and compounds that would make a modern chemist reach for protective gear. But here's the beautiful thing about chaos. It's also opportunity. All that volcanic activity wasn't just destructive, it was creative. Deep beneath the waves, where the ocean floor cracked and shifted, hydrothermal vents began to appear. These underwater geysers shot superheated, mineral-laden water into the surrounding sea, creating oases of chemical richness in an otherwise barren world. Imagine these vents as Earth's first laboratories. The water around them was warm, but not lethally hot. It was rich in hydrogen, carbon dioxide, and hydrogen sulfide, simple molecules that, under the right conditions, could link together in increasingly complex ways. The rocky surfaces around the vents provided templates and catalysts, helping molecules stick together and rearrange themselves into new configurations. Above ground, The atmosphere was performing its own chemical ballet. Lightning strikes, and there were many, provided bursts of energy that could forge simple organic compounds from the gases floating in the air. Methane and ammonia, when zapped with electricity, can produce amino acids, the building blocks of proteins. It's not unlike what Stanley Miller and Harold Urey discovered in their famous 1950s experiment, when they created a miniature version of early Earth in a laboratory flask. The planet's surface was a patchwork of extreme environments. Some areas were baked by intense heat. Others were frozen solid. Some were drenched in acid rain. Others were buried under layers of volcanic ash. But diversity, even harsh diversity, creates opportunities for chemistry to experiment. Molecules that couldn't survive in one environment might thrive in another. And then there was the gift from space. Those constant asteroid and comet impacts weren't just destructive. They were deliveries. Many of these cosmic visitors carried organic compounds, including amino acids, nucleotides, and other complex molecules. Each impact was like a care package from the universe, adding new ingredients to Earth's growing chemical cookbook. The early oceans also had something modern oceans lack: no life to compete with. Every chemical reaction had the luxury of proceeding without interference from hungry bacteria or metabolizing organisms. It was pure chemistry, unguided and uninterrupted, free to experiment with whatever combinations seemed most stable. Over millions of years, the planet began to calm down. The bombardment slowed. The atmosphere gradually shifted. The oceans cooled and deepened. Tidal pools formed along coastlines, creating shallow, sun-warmed laboratories where molecules could concentrate and interact. These pools would evaporate during the day, concentrating their chemical contents, then refill with fresh seawater, bringing new ingredients into the mix. It's hard to imagine such a violent, inhospitable world giving birth to something as delicate as life. But perhaps that's exactly what was needed. The harsh conditions weren't obstacles, they were the engine. The energy from lightning, volcanic heat, and cosmic impacts provided the power to build complex molecules. The chemical chaos created countless opportunities for new combinations to form. And somewhere in this maelstrom of rock and water, heat and electricity, simple molecules began to discover something remarkable. They could make copies of themselves. Not intentionally, of course. Chemistry doesn't have intentions. But certain arrangements of atoms turned out to be surprisingly good at attracting the right ingredients to build duplicates. This wasn't life yet, not as we'd recognize it. There were no cells, no DNA, no metabolism, just chemistry getting increasingly creative with itself. But it was the first tentative step on a journey that would eventually lead to butterflies and redwood trees, to symphonies and smartphones, to a species that could look back across billions of years and wonder at its own beginning. The primordial Earth was harsh, but it was also patient. It had all the time in the world to let chemistry experiment, to let molecules dance together in countless combinations until something special emerged. And in that patience, in that willingness to let chaos slowly organize itself into complexity, lay the seeds of everything that would follow. Life, it turns out, is surprisingly picky about its ingredients. While it's true that living things are made of the same basic elements found throughout the universe, carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur, the way these elements are arranged makes all the difference. It's like having flour, eggs, and sugar in your kitchen. You could make a cake, or you could make a mess. Life, fortunately, learned to make something far more elegant than either. Let's start with carbon, the star of the biological show. Carbon is life's favorite element because it's remarkably social. Each carbon atom can form 4 strong bonds with other atoms, creating complex, stable structures that can twist, branch, and link together like an intricate molecular sculpture. Silicon can do something similar, which is why science fiction writers love silicon-based aliens. But carbon does it better. It's lighter, more flexible, and forms stronger bonds. In the harsh conditions of early Earth, flexibility and strength were exactly what chemistry needed. The simplest carbon-based molecules that matter to life are amino acids. These are the building blocks of proteins, and they're surprisingly easy to make. Take some methane, ammonia, and water vapor, all abundant on early Earth. Add a spark of lightning, and voila, you get amino acids. It's like the universe's simplest recipe, requiring only basic ingredients and a good electrical storm. But amino acids alone don't make life. They're more like letters in an alphabet. To create meaning, you need to string them together into words, sentences, and eventually stories. In biological terms, you need to link amino acids into proteins. Proteins are the workhorses of life, folding into precise 3-dimensional shapes that allow them to grab onto other molecules, break chemical bonds, or speed up reactions. They're like molecular tools, each one designed for a specific job. Then there are nucleotides, the building blocks of DNA and RNA. These are more complex than amino acids, each one containing a sugar, a phosphate group, and one of 4 chemical bases. Think of them as more sophisticated letters in life's alphabet. When linked together, they can store information. The genetic code that tells a cell how to build proteins and, eventually, how to build more cells. Nucleotides are trickier to make than amino acids. They require more energy and more precise conditions. But early Earth had plenty of energy to spare, from lightning strikes to volcanic eruptions, to the steady bombardment of ultraviolet radiation from the unfiltered sun. Some scientists think nucleotides could have formed in warm little ponds that periodically dried up, concentrating the ingredients and providing the energy needed for complex chemistry. Another crucial ingredient is lipids, the molecules that eventually became cell membranes. Lipids are wonderfully schizophrenic. One end loves water, while the other end hates it. When you put lipids in water, they spontaneously arrange themselves into bubbles, with their water-loving heads facing outward and their water-hating tails tucked safely inside. These bubbles can enclose other molecules, creating tiny compartments where chemistry can happen without interference from the outside world. The beautiful thing about lipids is that they practically assemble themselves. Drop them in water, and they'll form membranes without any instruction manual. It's like having IKEA furniture that puts itself together, only much more reliable. This self-assembly property means that early Earth could have been dotted with countless tiny bubbles, each one a potential cradle for life. But how did these ingredients actually come together on early Earth? There are several competing theories, each more fascinating than the last. The classic approach is the primordial soup hypothesis, which imagines early oceans as a rich broth of organic molecules, slowly concentrating and complexifying over millions of years. Lightning and UV radiation provided the energy to forge new chemical bonds. while the vast timescales allowed for countless experiments in molecular combination. More recently, scientists have become enamored with hydrothermal vents as the cradle of life. These underwater hot springs create sharp gradients in temperature, pH, and chemical composition, perfect conditions for driving complex chemistry. The minerals around these vents can act as catalysts, speeding up reactions that might otherwise take millions of years. Some researchers think the very first metabolic pathways might have been powered by the chemical energy of these vents, long before life learned to harness sunlight. There's also the possibility that life's ingredients came from space. Meteorites regularly deliver organic compounds to Earth, including amino acids and even some nucleotide bases. The Murchison meteorite, which fell in Australia in 1969, contained over 70 different amino acids, many of which aren't found in terrestrial life. It's as if the universe had been practicing biochemistry long before Earth was ready for biology. Perhaps the most intriguing idea is that multiple environments contributed different pieces of the puzzle. Amino acids might have formed in the atmosphere, nucleotides in tidal pools, and lipids around hydrothermal vents. Over time, geological processes could have brought these ingredients together, like a cosmic mixing bowl stirring the components of life. What's remarkable is that all of these processes are still happening today. Organic molecules are still being created in the atmosphere during lightning storms. Hydrothermal vents still churn out complex chemistry. Meteorites still deliver extraterrestrial organics. The difference is that now, life itself dominates the planet's chemistry, making it much harder for new forms of life to get started. It's like trying to establish a new restaurant in a neighborhood that's already full of excellent eateries. The early Earth was a chemist's paradise, abundant raw materials, diverse energy sources, and unlimited time to experiment. No wonder it eventually stumbled upon the recipe for life. What's truly astonishing is that once chemistry discovered how to make copies of itself, it never looked back. Every protein in your body, every strand of DNA in your cells, traces its lineage back to those first tentative experiments in molecular self-assembly, conducted in the laboratories of an ancient alien Earth. So tonight, as you drift towards sleep, remember that you are made of stardust, yes, but more than that. You are made of molecules that learned to dance together billions of years ago, in oceans and pools and vents that no longer exist, under skies that no longer shine. You are chemistry that became curious about itself, matter that learned to dream. In the beginning was the Word, and the Word was probably RNA. This isn't meant to be theological, just biochemical. Of all the molecules that could have started life's story, RNA seems to be the most likely candidate for the starring role. It's a molecule that can do two things that are absolutely essential for life: store information and speed up chemical reactions. It's like a librarian who's also a master chef, keeping track of recipes while simultaneously cooking the meal. To understand why RNA is so special, we need to think about the chicken and egg problem that haunts the origin of life. Modern cells need DNA to store their genetic information, but they need proteins to read that DNA and carry out the cell's functions. But proteins are made according to instructions in DNA, and DNA can't be read without proteins. It's a circular dependency that seems impossible to break. How can you have one without the other? RNA solves this puzzle by being able to do both jobs, albeit not quite as well as DNA and proteins do their specialized tasks. RNA can store genetic information, just like DNA, The sequence of bases along an RNA molecule can encode instructions for building other molecules. But unlike DNA, which just sits there looking important, RNA can also fold into complex three-dimensional shapes that allow it to catalyze chemical reactions, just like proteins. These catalytic RNAs are called ribozymes, a portmanteau of ribonucleic acid, an enzyme. They were first discovered in the 1980s by Thomas Cech and Sidney Altman, who won the Nobel Prize for showing that RNA wasn't just a passive messenger between DNA and proteins, but an active participant in cellular chemistry. It was like discovering that the delivery person wasn't just bringing the mail, but was also secretly running the post office. The most famous ribozyme is found in the ribosome, the cellular machine that builds proteins. The heart of the ribosome, the part that actually links amino acids together, is made of RNA, not protein. This suggests that ribosomes are ancient molecular fossils, remnants of a time when RNA did most of the heavy lifting in cells. It's as if we found an old steam engine still chugging away inside a modern electric car. But if RNA was so talented, why did life eventually switch to the DNA and protein system we see today? The answer is specialization. DNA is much better at storing information because it's more stable than RNA. It has an extra chemical group that makes it less likely to fall apart, and it's usually found as a double helix, which provides backup copies of all information. RNA, on the other hand, is single-stranded and chemically finicky. It's like writing important documents on tissue paper instead of cardstock. Proteins, meanwhile, are much better catalysts than ribozymes. They can fold into a much wider variety of shapes. They're more stable, and they can be fine-tuned through evolution to be incredibly efficient. It's the difference between a Swiss Army knife and a full toolbox. The knife is handy when you need to do everything with one tool, but specialized tools work better for specific jobs. So the RNA world hypothesis suggests that early life went through an awkward teenage phase where RNA tried to do everything itself before eventually delegating tasks to more specialized molecules. DNA took over information storage, proteins took over most catalysis, and RNA settled into its current role as the messenger between them, though it still retains some of its ancient catalytic abilities. But how did this RNA world get started in the first place? That's where things get really interesting and really speculative. Making RNA from scratch requires linking together nucleotides, which are themselves complex molecules. Each nucleotide contains a sugar, a phosphate group, and one of 4 bases. Getting these components to assemble correctly under early Earth conditions is challenging, to put it mildly. One possibility is that simpler precursors came first. Maybe early replicators weren't made of RNA at all, but of simpler molecules that could copy themselves. These proto-genetic systems could have gradually evolved more complexity, eventually giving rise to RNA. It's like suggesting that the first writing systems weren't full alphabets, but simple pictographs that gradually became more sophisticated. Another idea is that RNA formation was helped along by mineral surfaces, particularly clays. Clay particles can act as templates, lining up nucleotides in the right orientation for linking. They can also concentrate nucleotides from dilute solutions, creating local environments where complex chemistry becomes more likely. It's a bit like having a patient chemistry teacher who holds your molecules in place while you figure out how to connect them. Some researchers have focused on specific sequences of RNA, that might have been particularly good at copying themselves. These are called ribozymes with replicase activity, RNA molecules that can catalyze the formation of more RNA. If such molecules existed in the RNA world, they could have kick-started a process of molecular evolution, with successful replicators becoming more common over time. The beauty of the RNA world is that it provides a plausible bridge between non-living chemistry and living biology. Once you have self-replicating RNA molecules, you have the basic requirements for evolution: heredity, variation, and selection. RNAs that are better at copying themselves will become more common. Random changes in sequence will create new variants, some of which might be even better replicators. Over time, this process could lead to increasingly sophisticated molecular systems. Evidence for the RNA world comes from several sources. The central role of RNA in modern cells suggests an evolutionary history where RNA was once more important. The fact that many essential cellular processes, from protein synthesis to gene regulation, depend on RNA hints at its ancient prominence. Even some of the most basic metabolic pathways involve RNA-based molecules like ATP and NAD, which could be molecular fossils from the RNA world. Perhaps most convincingly, scientists have been able to evolve RNA molecules in the laboratory that can do increasingly complex tasks. Starting with random sequences, researchers can select for RNAs that bind to specific targets, catalyze particular reactions, or even copy short pieces of other RNAs. While we haven't yet created a fully self-replicating RNA system in the lab, we're getting closer, and each experiment reveals new ways that RNA could have bootstrapped itself into existence. The RNA world hypothesis isn't proven, and it may never be. We're trying to reconstruct events that happened billions of years ago, leaving few traces behind. But it provides a compelling framework for thinking about how life could have emerged from non-life. It suggests that the transition wasn't a single miraculous event, but a gradual process of increasing molecular sophistication. Tonight, as your own RNA molecules busily carry messages between your genes and your ribosomes, remember that they're part of an ancient lineage stretching back to the very dawn of life. Every protein being made in your cells right now is being assembled by ribosomes that contain echoes of that primordial RNA world, where molecules first learned the trick of making copies of themselves, setting the stage for the magnificent complexity of life we see today. At some point, life decided it needed walls, not for privacy exactly, but for something even more fundamental: the ability to control what came in and what went out. Up until this moment in our story, all the fascinating chemistry we've been describing was happening in the open ocean, in tidal pools, around hydrothermal vents, essentially in public. And while public chemistry can accomplish remarkable things, private chemistry, it turns out, can accomplish even more. The problem with doing biochemistry in the open ocean is the same problem you'd have trying to bake a cake in a swimming pool. Everything gets diluted. The moment you create an interesting molecule, it disperses into the vast surrounding water, becoming so spread out that it can barely find other molecules to react with. It's like trying to have a conversation in a football stadium. Possible, but not ideal. What early life needed was a way to concentrate its chemistry, to keep useful molecules close together, while keeping harmful ones at bay. The solution was elegantly simple: build a membrane. Not a rigid wall like a fortress, but a flexible boundary like a soap bubble. And just as soap bubbles form naturally when you blow through a soapy film, the first cell membranes may have formed spontaneously from molecules that were already floating around in the primordial soup. These membrane-forming molecules are called phospholipids, though the early versions were probably simpler than what we see in modern cells. Each molecule has a peculiar personality. One end loves water, while the other end despises it. When you put these schizophrenic molecules in water, they naturally arrange themselves to keep their water-hating tails away from the wet environment. The easiest way to do this is to form a double layer, with all the water-loving heads facing outward toward the water, and all the water-hating tails tucked safely inside, facing each other. This arrangement creates a membrane that's simultaneously strong and flexible. It's strong enough to maintain a boundary between inside and outside, but flexible enough to bend, stretch, and even heal small tears. It's like having a wall made of liquid, substantial enough to keep things separate, but dynamic enough to change shape when needed. But here's where it gets really clever. These membranes aren't just passive barriers, they're selective barriers. Small molecules like water can slip through relatively easily, while larger molecules like proteins and nucleic acids are kept inside. It's like having a bouncer who lets in friends but keeps out troublemakers. Except the bouncer is made of molecules and operates according to the laws of chemistry, rather than personal preference. The first cells, or protocells, as scientists call them, were probably just these simple membrane bubbles, with some interesting chemistry trapped inside. Imagine a soap bubble filled with RNA molecules, amino acids, and other organic compounds. The membrane would concentrate these molecules, allowing them to interact more frequently and efficiently than they could in the open ocean. Chemical reactions that might take millions of years in dilute seawater could happen in minutes or hours inside a protocell. These early cell membranes had another crucial property. They could grow and divide. When a membrane bubble gets too large, it becomes unstable and spontaneously splits into smaller bubbles. Each daughter bubble inherits some of the parent's internal contents. If those contents included self-replicating RNA molecules, then cell division would also mean genetic reproduction. The RNA gets copied, the cell grows larger, the membrane divides, and suddenly you have two cells where there was once one. This is where things start to resemble what we'd recognize as life. You have information storage in the RNA, metabolism, chemical reactions inside the cell, and reproduction when the cell divides. These are often considered the three essential features of life, though philosophers and biologists still debate the exact definition of what it means to be alive. The transition from free-floating molecules to enclosed cells was probably gradual. Early membranes might have been leaky and unstable, forming and dissolving like soap bubbles in a sink. But over time, evolution would have favored cells with better membranes, ones that lasted longer, protected their contents more effectively, and divided more reliably. Natural selection, that patient sculptor of complexity, began to refine the first crude attempts at cellular life. Modern cell membranes are marvels of biochemical engineering. They contain dozens of different types of lipids, each contributing specific properties to the membrane's behavior. They're studied with protein channels and pumps that carefully control what enters and exits the cell. They can bend and flex without breaking, merge with other membranes, and even repair damage to themselves. They're like smart walls that know exactly what they're protecting and why. But those early protocells were probably much simpler. Their membranes might have been made of just one or two types of lipids. They might not have had sophisticated transport proteins. They were probably fragile and short-lived by modern standards. But they represented a crucial step in life's journey, the transition from chemistry happening in the environment to chemistry happening in a controlled, protected space. The formation of the first cells also solved another crucial problem: how to maintain favorable conditions for life. The early ocean was a chaotic place with wildly varying temperature, pH, and chemical composition. But inside a cell, conditions could be buffered and controlled. If the outside environment became too acidic, the cell could maintain a more neutral pH inside its membrane. If nutrients became scarce outside, the cell could concentrate them inside. This ability to create and maintain internal conditions different from the external environment is called homeostasis, and it's one of the defining features of life. Every cell in your body, from your neurons to your muscle fibers, is constantly working to maintain stable internal conditions, despite the changing environment outside. It's like having a personal climate control system that's been running continuously for billions of years. The first cells also opened up new possibilities for evolution. Once genetic material was enclosed within membranes, it became possible for different cells to develop different internal chemistries. Some cells might become better at harvesting energy, while others might become better at building complex molecules. Over time, this could lead to specialization and, eventually, to the cooperation between different types of cells that we see in multicellular organisms. Looking back, the evolution of the first cells seems almost inevitable. Given molecules that can form membranes and chemistry that can benefit from being concentrated, the formation of protocells becomes not just possible, but probable. It's one of those beautiful examples of how complexity can emerge from simplicity through the patient workings of physical and chemical laws. Tonight, as you settle into sleep, consider that every cell in your body is a direct descendant of those first brave bubbles that dared to wall themselves off from the ancient ocean. The membrane around each of your cells carries on a tradition that began billions of years ago, when chemistry first learned to build itself a private workshop where the real magic of life could begin. Being alive, as it turns out, is exhausting work. Every second of every day, your cells are bustling with activity, building proteins, copying DNA, moving materials around, responding to signals, and generally maintaining the incredible complexity that keeps you breathing, thinking, and dreaming. All of this activity requires energy, and lots of it. In fact, you burn through roughly your own body weight in ATP, the cell's energy currency, every single day. That's a lot of molecular fuel for what seems like the simple act of existing. But where does all this energy come from? Today, most life on Earth ultimately depends on the sun. Plants capture solar energy through photosynthesis. Animals eat plants or other animals, and the energy gets passed around the food web like a never-ending game of energetic hot potato. But 4 billion years ago, when life was just getting started, photosynthesis didn't exist yet. The sun was dimmer, the atmosphere was opaque, and there weren't any plants to capture light anyway. So how did the first living things power themselves? The answer lies in chemistry itself. Long before life learned to harness sunlight, it discovered that certain chemical reactions could provide energy. These reactions don't require any external energy source. They happen spontaneously when the right molecules encounter each other under the right conditions. It's like finding out that you can power your house by mixing baking soda and vinegar, except the chemistry is more sophisticated and the house is a living cell. The most likely place for this chemical energy harvesting to begin was around hydrothermal vents on the ocean floor. These underwater geysers create a perfect environment for energy-releasing chemistry. Hot, mineral-rich water gushes up from deep within the earth, carrying hydrogen sulfide, hydrogen gas, and various metals. When these chemicals encounter the cooler, more oxidized seawater, they react eagerly, releasing energy in the process. Modern organisms that live around hydrothermal vents still use this ancient strategy. They're called chemosynthetic organisms, and they can derive energy from reactions involving hydrogen sulfide, methane, ammonia, or hydrogen gas. These organisms form the base of deep-sea food webs, supporting entire ecosystems in the eternal darkness of the ocean depths. They're like underwater solar panels, except they harvest chemical energy instead of light. But early life couldn't just use energy willy-nilly. Raw chemical energy is like trying to power a delicate watch with a lightning bolt. Too much, too fast, and you'll fry everything. What life needed was a way to capture energy from chemical reactions and store it in a more manageable form. This is where metabolism comes in, the intricate network of chemical reactions that breaks down molecules to release energy and builds up molecules to store it. The earliest metabolic pathways were probably very simple compared to what we see in modern cells. Instead of the elegant, multi-step processes that characterize modern metabolism, early life might have relied on just a few key reactions. These reactions had to accomplish two essential tasks: break down available molecules to release energy, and use that energy to build the complex molecules needed for growth and reproduction. One of the most ancient metabolic pathways still used by life today is called glycolysis. It breaks down sugar molecules to release energy, and it doesn't require oxygen. A good thing, since there wasn't any free oxygen in the early atmosphere. Glycolysis can happen in almost any cell, under almost any conditions. It's like the metabolic equivalent of a reliable old truck that runs on whatever fuel you put in the tank. Another ancient pathway involves the breakdown of organic acids like acetate. This process, called acetogenesis, can provide energy while also producing the building blocks for other important molecules. Some scientists think acetogenesis might have been one of the very first metabolic pathways, emerging in the chemical gardens around hydrothermal vents where organic acids would have been abundant. But metabolism isn't just about breaking things down for energy. Life also needs to build things up: proteins, nucleic acids, membranes, and all the other molecular machinery that makes a cell work. This constructive side of metabolism is called anabolism, and it requires energy input rather than producing it. It's like the difference between burning wood for heat and growing a tree. Both are important, but they operate in opposite directions. The genius of metabolism is how it links these two processes together. The energy released by breaking down molecules is captured in the special energy storage molecules like ATP. This stored energy can then be used to power the construction of new molecules when and where they're needed. It's like having a rechargeable battery that gets refilled by burning fuel and can then power whatever devices you need to run. ATP, adenosine triphosphate, is so central to life that it's sometimes called the universal energy currency. Every known form of life uses ATP to store and transport energy within cells. The molecule itself is relatively simple. It contains an adenine base, a ribose sugar, and 3 phosphate groups linked together. When one of those phosphate bonds is broken, energy is released. When the bond is reformed, energy is stored. It's molecular banking at its finest. Interestingly, ATP contains ribose, the same sugar found in RNA. This isn't a coincidence. Many of the molecules involved in energy metabolism contain components that are also found in genetic material. This suggests that metabolism and genetics co-evolved, each one depending on the other from very early in life's history. It's like discovering that the engine and transmission in your car were designed by the same team of engineers. The evolution of metabolism also required the development of enzymes, protein catalysts that speed up chemical reactions. Without enzymes, most metabolic reactions would happen so slowly that life would grind to a halt. But with enzymes, reactions that might take years to complete on their own can happen in milliseconds. Early life probably used ribozymes, RNA molecules with catalytic activity, before proteins took over most enzymatic functions. As metabolism became more sophisticated, it also became more efficient. Early metabolic pathways probably wasted a lot of energy, like a leaky fuel tank. But evolution gradually selected for more efficient processes, ones that could extract more energy from the same starting materials or accomplish the same biosynthetic goals with less energy expenditure. Over millions of years, metabolism became a finely tuned engine capable of supporting increasingly complex forms of life. The development of cellular respiration, the process that uses oxygen to extract energy from glucose, was a major evolutionary breakthrough, but it came much later in our story. For the first billion or more years of life's existence, metabolism operated without oxygen, using the chemical energy available in the ancient environment. These anaerobic metabolic pathways are still used by many organisms today, particularly those that live in oxygen-free environments like deep sediments, the guts of animals, or the same hydrothermal vents where metabolism might have first evolved. What's remarkable is that all the complexity of modern metabolism, the hundreds of interconnected pathways that keep your cells running, can be traced back to these simple beginnings. Every breath you take, every heartbeat, every thought is powered by metabolic processes that have their roots in the chemical gardens of the early Earth. Metabolism is life's oldest profession, the fundamental skill that transformed chemistry into biology and made everything else possible. So tonight, as your own metabolism quietly continues its ancient work, converting the food you ate into the energy that maintains your body and mind, remember that you're participating in a process that's been refined over billions of years. Your cells are running on the same basic principles that powered the very first living things, updated and optimized through countless generations, but still recognizably connected to those pioneering experiments in cellular chemistry that took place in an alien ocean under alien skies, so long ago that the very rocks have forgotten. RNA was brilliant, but it wasn't built to last. For all its talents, storing information, catalyzing reactions, serving as life's original Swiss Army knife, RNA has one significant flaw. It's chemically fragile. RNA molecules have a tendency to fall apart, especially in the warm, salty conditions where early life was trying to establish itself. It's like trying to write your memoirs on tissue paper in a rainstorm. Possible, but not ideal for long-term storage. This instability created a problem for evolving life. As organisms became more complex, they needed to store more information. Instructions for building proteins, regulations for when to make them, and coordination signals for increasingly sophisticated cellular processes. But RNA's fragility limited how much information could be reliably maintained from one generation to the next. Imagine trying to run a library where the books randomly lost pages, or a computer where the hard drive occasionally deleted files. It works for a while, but it's not a sustainable long-term strategy. Enter DNA, deoxyribonucleic acid, RNA's more stable cousin. The difference between RNA and their DNA might seem minor. DNA is missing just one oxygen atom per sugar molecule, and it uses thymine instead of uracil as one of its 4 bases. But these small chemical changes have enormous consequences. That missing oxygen atom makes DNA much less reactive, much less likely to break down spontaneously. It's the molecular equivalent of switching from tissue paper to cardstock. Even more importantly, DNA almost always exists as a double helix, 2 complementary strands wound around each other like a twisted ladder. This double-stranded structure provides built-in redundancy. If one strand gets damaged, the information can be recovered from the other strand. It's like keeping 2 identical copies of every important document, filed in separate, secure locations. The transition from an RNA-based genetic system to a DNA-based one wasn't sudden. It probably happened gradually, with early organisms using both RNA and DNA for different purposes. DNA took over the job of long-term information storage, while RNA continued to serve as a messenger, carrying copies of genetic instructions from the DNA archives to the protein-building machinery. This division of labor is still how modern cells operate, with DNA serving as the master library and RNA as the circulation system. But why did life bother to evolve DNA at all? Why not just make RNA more stable? The answer probably has to do with the trade-off between stability and activity. RNA's instability is actually part of what makes it useful as a catalyst and regulatory molecule. Its flexibility allows it to fold into complex shapes and change conformation in response to environmental conditions. DNA, being more stable, is less flexible and therefore less suitable for catalytic roles. It's the difference between a sturdy filing cabinet and a nimble acrobat. Each is perfect for its job, but they're not interchangeable. The evolution of DNA also coincided with another major innovation: the development of sophisticated DNA repair mechanisms. Modern cells have dozens of different systems for detecting and fixing DNA damage. Some repair systems fix spelling errors, some fix structural damage, and some can even patch together a shattered chromosome from fragments. These repair systems are so effective that the human genome, despite being constantly bombarded by radiation and chemical damage, maintains its integrity across decades of cellular divisions. With stable DNA storage and robust repair mechanisms, life could suddenly afford to be much more complex. Genomes could grow larger, storing instructions for hundreds or thousands of different proteins. Regulatory networks could evolve, allowing cells to respond to environmental changes with sophisticated programs of gene expression. The genetic code itself could be optimized, with redundancy built in to minimize the impact of mutations. This transition also enabled the evolution of more complex cellular structures. Modern cells contain dozens of different organelles and molecular machines, each built according to genetic instructions stored in DNA. The ribosome alone, the cellular machine that translates genetic information into proteins, is made of more than 50 different components, all encoded in DNA. Building such intricate machinery requires genetic instructions that are both detailed and reliable, qualities that RNA simply couldn't provide at the necessary scale. The DNA revolution also transformed how evolution itself works. With stable genetic storage, mutations became less random and more meaningful. Instead of genetic information constantly degrading, changes in DNA could accumulate gradually, allowing natural selection to fine-tune organisms over many generations. It's the difference between trying to evolve in a house of cards versus building on a solid foundation. The stable foundation allows for much more sophisticated architecture. Interestingly, the evolution of DNA probably required its own evolutionary chicken and egg problem. DNA is more stable than RNA, but it's also more difficult to synthesize. Making DNA requires specialized enzymes that are themselves encoded in genetic material. How do you bootstrap a DNA-based genetic system when you need DNA to make the enzymes that make DNA? The answer probably lies in the gradual coevolution of DNA synthesis machinery and DNA storage systems, each improvement enabling the next step in complexity. The central dogma of molecular biology, DNA makes RNA makes protein, emerged from this evolutionary transition. DNA serves as the permanent archive, storing genetic information in a stable, easily replicable form. RNA serves as the working copy, carrying specific genetic messages from the archive to the protein synthesis machinery. Proteins serve as the molecular workforce, carrying out the actual functions of the cell according to the instructions copied from DNA. This system is remarkably robust and flexible. The same genetic information can be expressed differently in different cell types, allowing a single genome to specify many different kinds of cells. Genes can be turned on or off in response to environmental conditions. Multiple RNA copies can be made from a single gene, amplifying the cellular response when needed. It's like having a master blueprint that can be photocopied, edited, and distributed throughout a vast construction project. The evolution of DNA also enabled the eventual development of sexual reproduction, which requires the precise pairing and exchange of genetic material between individuals. RNA-based genetics would have been too error-prone for this process to work reliably. DNA stability and the sophisticated machinery for DNA repair and recombination made sexual reproduction possible, which in turn dramatically accelerated the pace of evolution by creating new combinations of genes in each generation. Modern DNA is remarkably similar across all forms of life. The genetic code, the system that translates DNA sequences into protein sequences, is nearly universal, used by bacteria, plants, animals, and every other form of life on Earth. This universality suggests that the DNA-based genetic system evolved once and then spread to all subsequent life forms. It was such a successful innovation that once it emerged, it took over completely. Yet DNA still carries traces of its RNA ancestry. The enzymes that copy DNA, DNA polymerases, can only add nucleotides to existing RNA primers. It's as if modern DNA replication still needs a little help from its older sibling to get started. Many other aspects of DNA metabolism also depend on RNA, suggesting that the transition from RNA to DNA was more of a partnership than a replacement. Tonight, as your cells carefully maintain and repair the 3 billion base pairs of DNA in your genome, remember that you're the beneficiary of one of evolution's greatest innovations. The stable storage of genetic information in DNA made possible all the complexity of multicellular life, from the intricate development programs that built your brain to the sophisticated immune system that protects you from disease. Every cell division, every breath, every dream depends on the reliable inheritance of genetic information made possible by DNA's molecular revolution, an innovation so successful that once it emerged, life never looked back. Once life learned the rules, it immediately started breaking them. Having established the basic principles of existence, information storage, energy metabolism, cellular compartments, and reproduction, life embarked on what can only be described as an experimental binge, like a child who has just mastered walking and immediately wants to try skipping, jumping, and possibly climbing the curtains. Early life began exploring every possible variation on the theme of being alive. This diversification didn't happen overnight. We're talking about hundreds of millions of years of patient tinkering, trial and error, and gradual refinement. But the results were extraordinary. From a single ancestor, or possibly a small community of closely related early organisms, life branched out into forms so different that they might as well have been living on different planets. Some learned to eat sunlight, others thrived in boiling water. Still others figured out how to breathe sulfur instead of oxygen. It was biology's greatest period of experimentation, a time when anything seemed possible, and much of it actually was. The first major split in the tree of life separated what we now call bacteria and archaea. At first glance, these two groups look remarkably similar. Both are single-celled organisms without nuclei, both are microscopic, and both can be found in similar environments. For decades, scientists lumped them together as prokaryotes, cells without nuclei, in contrast to eukaryotes like plants, animals, and fungi, whose cells have nuclei and other membrane-bound organelles. But appearances can be deceiving. When scientists began studying the molecular machinery of bacteria and archaea in detail, they discovered profound differences. The molecules that bacteria use to build proteins are fundamentally different from those used by archaea. Their cell walls are constructed from entirely different materials. Even their DNA is organized and processed in different ways. It's like discovering that two cars that look identical from the outside actually have completely different engines, transmissions, and electrical systems. These differences suggest that bacteria and archaea diverged very early in life's history, possibly more than 3 billion years ago. They represent two fundamentally different solutions to the problem of how to be a single-celled organism. Neither approach is inherently better than the other. They're just different ways of accomplishing the same basic goals: gathering energy, reproducing, and surviving in a challenging world. Bacteria, as a group, became masters of metabolic diversity. They learned to extract energy from an astonishing variety of sources. Some bacteria harvest energy from sunlight, like tiny solar panels. Others derive energy from chemical reactions involving sulfur, nitrogen, iron, or dozens of other elements. There are bacteria that can live on pure hydrogen, bacteria that breathe rock instead of oxygen, and bacteria that can survive radiation levels that would sterilize a hospital operating room. This metabolic flexibility allowed bacteria to colonize virtually every environment on Earth. They live in hot springs where the water temperature exceeds the boiling point. They thrive in Antarctic ice where nothing has moved for millions of years. They float in clouds, swim in underground rivers, and form vast communities on the ocean floor. Some bacteria can survive in space, protected only by a thin biofilm. If aliens were to visit Earth and sample its life forms, they would likely conclude that this is a planet dominated by bacteria, and they would be right. Archaea, meanwhile, became the extremophiles of the microbial world. While bacteria certainly include some hardy species, Archaea seem to specialize in living in conditions that would challenge a science fiction writer's imagination. They flourish in salt concentrations that would preserve a mummy. They grow in acid strong enough to dissolve metal. They produce methane in the guts of termites and cows, generate energy in boiling underwater vents, and somehow manage to reproduce in environments so hostile that early researchers called them archaebacteria, ancient bacteria that must represent life's earliest forms. We now know that archaea aren't necessarily more ancient than bacteria, despite their name. They're just specialized for extreme conditions. This specialization might reflect their evolutionary history. Perhaps early Earth was so harsh that only extremophile organisms could survive, and archaea are the descendants of those hardy pioneers. Or perhaps archaea simply evolved different biochemical solutions that happen to work well under extreme conditions. Either way, they represent a fascinating alternative approach to single-celled life. But the really dramatic diversification came with the evolution of different metabolic strategies. Early life was probably heterotrophic, meaning it obtained energy by consuming organic molecules that were already present in the environment. This is like living off your savings account. It works for a while, but eventually you run out of money. The early ocean contained a rich supply of organic compounds, but it wasn't infinite. Life needed to find renewable sources of energy. The solution was to learn how to make food rather than just consuming it. This led to the evolution of autotrophy, the ability to build organic molecules from simple inorganic compounds. The most famous form of autotrophy is photosynthesis, which uses sunlight to convert carbon dioxide and water into glucose. But photosynthesis wasn't the first autotrophic process to evolve. That honor probably goes to chemosynthesis, which derives energy from chemical reactions rather than sunlight. Chemosynthetic organisms are still thriving today, particularly around hydrothermal vents and in other environments where chemical energy is abundant. They form the base of ecosystems that exist in complete darkness, powered entirely by Earth's internal chemistry. These organisms might give us hints about what early autotrophic life looked like before photosynthesis illuminated the biological world. The evolution of different metabolic strategies also led to the first ecological relationships between different organisms. Some bacteria began producing waste products that other bacteria could use as food. Others developed mutually beneficial partnerships, sharing resources and protection. Still others became predators, developing ways to hunt and consume their microscopic neighbors. The microbial world became an ecosystem, with producers, consumers, and decomposers all playing their parts in the cycling of energy and materials. This period also saw the evolution of increasingly sophisticated cellular machinery. Early ribosomes became more efficient at building proteins. DNA replication became more accurate. Cell membranes became more selective about what they allowed to pass through. Many of the molecular machines that power modern cells were invented during this period of rapid evolutionary experimentation. Perhaps most importantly, life learned how to deal with its own success. As organisms became more numerous and more efficient, they began to change their environment. Waste products accumulated, resources became scarce, competition intensified. Life had to evolve not just to survive in the natural environment, but to survive in an environment increasingly shaped by life itself. This co-evolution of life and environment set the stage for one of the most dramatic events in Earth's history, the Great Oxygenation Event. But that's a story for our next chapter. For now, it's enough to marvel at the extraordinary creativity of early evolution, the way life took a simple set of rules and used them to generate an almost infinite variety of solutions to the challenge of existence. The diversification of early life wasn't just about survival. It was about exploration. Each new metabolic pathway, each new cellular innovation, each new ecological strategy opened up previously unimaginable possibilities. Life wasn't content to simply exist. It wanted to exist in every conceivable way. And in doing so, it transformed a sterile planet into a living world, setting the stage for all the complexity and beauty that would follow. Tonight, as you rest, remember that you carry within you the molecular legacy of this great diversification. Your cells use metabolic pathways that were invented billions of years ago by bacteria you'll never see. Your DNA is organized according to principles that were established in the earliest days of cellular life. You're the descendant of organisms that dared to break the rules, to try new approaches, to explore the vast space of biological possibility. In your very existence, the experimental spirit of early life lives on. Sometimes the most revolutionary changes come from the smallest rebels. Around 2.4 billion years ago, a group of microscopic organisms began doing something that had never been done before on Earth. They started splitting water molecules and releasing oxygen gas as a waste product. These tiny revolutionaries were cyanobacteria, and they were about to change the world in ways that would make every subsequent political revolution look like a minor disagreement over tea preferences. At the time, this new process, which we now call oxygenic photosynthesis, probably seemed like just another metabolic innovation. Life had already invented many ways to capture energy from the environment. Some organisms use chemical energy from hydrothermal vents. Others had learned to harvest sunlight using simpler forms of photosynthesis that didn't produce oxygen. The cyanobacteria's version was just one more entry in life's growing catalogue of energy harvesting strategies. No one could have predicted that this seemingly minor innovation would fundamentally reshape the planet. The key breakthrough was figuring out how to use water as an electron donor in photosynthesis. Previous forms of photosynthesis had relied on compounds like hydrogen sulfide or organic molecules as electron sources. These worked fine, but they were limited in availability. Water, on the other hand, was everywhere. If you could crack the code for splitting water molecules, you would have access to an essentially unlimited supply of electrons for photosynthesis. It was like discovering how to run your car on seawater instead of gasoline. But there was a catch. Splitting water requires a lot of energy, more than simple sunlight can provide directly. The cyanobacteria had to evolve a complex, two-stage photosynthetic system that could capture and concentrate light energy effectively enough to break the strong chemical bonds holding water molecules together. This system, called photosystem 2, is one of the most sophisticated molecular machines ever evolved. It uses quantum mechanical effects to capture light with near-perfect efficiency and channels that energy into the water-splitting reaction. The byproduct of this process was oxygen gas. And that's where things got interesting, in the same way that a small leak in a dam can get interesting. At first, the oxygen produced by cyanobacteria was probably absorbed by iron and other reduced minerals in the ocean. These minerals acted like chemical sponges, soaking up the oxygen before it could accumulate in the atmosphere. But eventually, the oxygen production exceeded the absorption capacity of these mineral sponges. Oxygen began to bubble up through the ocean and escape into the atmosphere. This was catastrophic for most existing life forms. Oxygen is highly reactive. It's what makes fire possible, what causes metal to rust, what turns apples brown when you leave them exposed to air. For organisms that had evolved in an oxygen-free world, the rising oxygen levels were like being slowly poisoned. Oxygen damaged their cell membranes, interfered with their enzymes, and generally made life miserable. Many species went extinct during this period, which scientists somewhat dramatically call the Great Oxygenation Event, though oxygen catastrophe might be more accurate from the perspective of the victims. But catastrophe for some became opportunity for others. A few organisms managed to survive the rising oxygen levels, and some even learned to turn this reactive gas to their advantage. They evolved cellular respiration, the process that uses oxygen to extract energy from organic molecules with remarkable efficiency. Oxygen-based respiration can extract roughly 18 times more energy from glucose than the anaerobic processes that preceded it. It was like upgrading from a bicycle to a jet engine. This efficiency boost had profound consequences. With access to much more energy, life could afford to be more complex. Cells could grow larger, support more sophisticated internal structures, and engage in more energy-intensive activities. The stage was set for the evolution of eukaryotic cells, the complex cells with nuclei and organelles that eventually gave rise to plants, animals, and fungi. The Great Oxygenation Event also transformed Earth's atmosphere in other ways. Oxygen in the upper atmosphere was converted to ozone by ultraviolet radiation from the sun. This ozone layer began to shield the planet's surface from harmful UV rays, making it safer for life to venture into shallow water and eventually onto land. It's ironic that the same process that poisoned much of early life also created the protective barrier that would allow life to flourish in new environments. The event also affected Earth's climate. Oxygen reacted with methane in the atmosphere, removing this potent greenhouse gas and causing global temperatures to drop. Some scientists think this led to a global ice age that lasted for millions of years, one of the first times that life's activities significantly altered the planet's climate. It wouldn't be the last. Perhaps most importantly, the Great Oxygenation Event established oxygen as a permanent feature of Earth's atmosphere. Once oxygen levels stabilized at roughly their current levels, they created a new set of environmental conditions that would persist for billions of years. This stable, oxygenated environment became the backdrop for all subsequent evolution on Earth. The cyanobacteria responsible for this transformation are still with us today. They continue to produce much of the oxygen we breathe, both in the oceans and in the freshwater environments around the world. Every breath you take contains oxygen molecules that were produced by the descendants of those first water-splitting pioneers. In a very real sense, we owe our existence to these microscopic organisms and their revolutionary approach to harvesting sunlight. The chloroplasts in plant cells, the organelles that carry out photosynthesis in trees, grasses, and algae, are actually descendants of ancient cyanobacteria that were engulfed by early eukaryotic cells. This process, called endosymbiosis, allowed the efficiency of cyanobacterial photosynthesis to be combined with the complexity of eukaryotic cells. The result was plants, which took photosynthesis from the ocean to the land and eventually covered much of the planet's surface with green. Looking back, the Great Oxygenation Event represents a perfect example of how life doesn't just adapt to its environment. It actively transforms that environment, often in ways that make further evolution possible. The cyanobacteria didn't set out to create an oxygen-rich atmosphere. They were simply trying to make a living in a competitive world. But their metabolic innovation had consequences that reached far beyond their immediate needs. This pattern, life changing the environment, which then shapes the course of future evolution, has been repeated many times throughout Earth's history. The evolution of trees changed the composition of soils and the patterns of water flow. The evolution of animals with shells and skeletons altered the chemistry of the oceans. The evolution of humans is currently changing the composition of the atmosphere, the distribution of species, and the very face of the planet. Each of these transformations opens new possibilities while closing others. The Great Oxygenation Event made complex, multicellular life possible, but it also eliminated many of the metabolic pathways that had flourished in the oxygen-free world of early Earth. Evolution is not just about organisms adapting to fixed environments. It's about the co-evolution of life and environment, each shaping the other in an endless dance of change and adaptation. Tonight, as you breathe the oxygen-rich air that surrounds you, remember that each breath connects you to the most successful environmental revolution in Earth's history. The oxygen in your lungs was produced by organisms not much bigger than the bacteria living in your gut. Yet their collective impact was so profound that it literally changed the color of the sky. You exist because billions of years ago, tiny cyanobacteria learned to split water and happened to be very good at it. Your every breath is a reminder that the smallest organisms can have the largest consequences, that innovation often comes from unexpected sources, and that the air you breathe is itself a gift from life's grand experiment with existence. The story of early life's diversification and the Great Oxygenation Event reminds us that life is not just about individual survival and reproduction. It's about transformation, of molecules into cells, of cells into ecosystems, of ecosystems into planets capable of supporting unimaginable complexity and beauty. And that transformation continues today, with every breath, every heartbeat, every dream, adding new chapters to the longest and most extraordinary story ever told, the story of life itself. And so, dear friend, we arrive at the end of our gentle journey through deep time, having wandered from sterile chemistry to the first stirrings of life, from simple molecules to the complex cells that eventually learn to dream. We've seen how Earth's earliest rebels, tiny bacteria splitting water in ancient seas, gave us the very air we breathe. We've traced the long path from RNA's first tentative experiments in self-copying to the sophisticated genetic systems that guide every heartbeat and help every flower bloom. What a curious thing to realize that every cell in your body carries within it the accumulated wisdom of 4 billion years of trial and error, success and failure, innovation and persistence. The mitochondria powering your thoughts were once independent bacteria. The genes directing your development were refined across countless generations. Even your ability to breathe oxygen, something so automatic you rarely think about it, depends on molecular machinery perfected by organisms you'll never see, in oceans that no longer exist, under skies that have long since changed. The question we set out to answer, how did life actually begin on Earth, turns out to have been the perfect question for bedtime. Not because the answer is simple, but because it reminds us that existence itself is the most remarkable thing of all. You are here not by accident, but as the result of the most extraordinary chain of innovations, adaptations, and lucky breaks in the known universe. You are chemistry that learned to wonder about itself, matter that became curious about its own origins. If you found any peace in this quiet expedition through time, if the wonders of early life helped ease you towards sleep, feel free to follow us for more journeys through science in the future. Because while we may have reached the end of life's beginning, the story itself is far from over. It continues in every breath, every dream, every moment of wonder you'll experience as you drift off into the gentle darkness. Thanks for joining us here at Sleep Science. Sleep well, knowing that in your very existence, the miracle of life's first moments lives on.